to our TED Talk. Today we will be talking about a form of blindness that can be eradicated using genetic modification. Using CRISPR, we will now be able to cure Leber Congenital Amaurosis, which is a genetic disorder that results in blindness. LCA is a disorder that occurs in 1 in every 40,000 newborns. LCA is one of the main causes of blindness among people across the world. Considering there are 285 million blind people worldwide, curing LCA would be revolutionary. Since people who have LCA lose their vision, their lives become significantly more difficult. These people will always be forced to be dependent on others, making their lives very hard. Luckily, by using CRISPR, LCA can be cured.
Blind people and the parents of children who suffer from LCA would be the stakeholders to our cause. People who suffer from this disease would no longer need to live life without vision with the assistance of CRISPR. When a child gets LCA, CRISPR would be able to target and change the mutated genes, allowing the child to keep their vision before it goes away completely. Additionally, this would greatly improve the lives of the parents, who would no longer need to deal with the added responsibilities of raising a blind child. Additionally, this would interest doctors, as they'd be the ones who would prescribe the CRISPR solution to a patient.
According to the U.S. National Library of Medicine, Leber Congenital Amaurosis can result from mutations in at least 14 genes in the body, all of which are necessary for normal vision. These known genes that cause the disorder include CRX, IMPDH1, IQCB1, LCA5, LRAT, NMNAT1, RD3, RDH12, RPGRIP1, SPATA7, and TULP1. While all of these genes control the production of proteins necessary for normal vision, the lack of any of these can result in LCA.
Genetic engineering is a complex process that occurs when a piece of genetic code is targeted in an organism and is either taken out of the system or placed into the code. In our case, we need to engineer a method to export the mutated gene out of the code and replace it with a normal functioning gene. Currently, there is one main use of the technology that can alter genes. This is called CRISPR-Cas9, and it is a technological piece of equipment that can be used to alter DNA sequences and the function of genes. The Cas9 part of the DNA is used for cutting specific parts of DNA. The CRISPR-Cas9 cuts out a specific gene, whichever one is decided by the user, and then has it replaced with a new healthy gene. In our case, any of the 16 known mutated genes that cause LCA can be fixed. This technology is very advanced, and it can be used to help fix the genetic disorder of blindness from Leber Congenital Amaurosis.
DNA and chromosomes play a role that is essential to making life. DNA stems from the chromosomes of an organism's parents, with half of each parent's DNA being transferred to the new organisms. These chromosomes combine to make new DNA, which acts as a code for traits in an organism to be established. The DNA creates proteins that create traits and other characteristics in an organism. LCA occurs when a mutated gene from a parent's chromosomes are passed on to their offspring. In the case of LCA, the trait is recessive, meaning it is only expressed in an organism if a mutated gene is inherited from both parents.
The main genes that become mutated include CEP290, CRB1, GUCY2D, and RPE65. While those are the main genes that become mutated, errors in the AIPL1, CRX, IMPDH1, IQCB1, LCA5, LRAT, NMNAT1, RD3, RDH12, RPGRIP1, SPATA7, and TULP genes can also be the cause of LCA. Here's a breakdown of what all the different genes do.
The first gene is AIPL1, which is the gene responsible for the photoreceptor pineal express gene that encodes aryl hydrocarbon interacting proteins. The next gene is CEP290, which plays a role in the creation of cell structures called centrosomes in cilia, which help with the senses such as smell, hearing, and sight.
Moving on, the next gene is CRB1, which is part of the development in photoreceptor cells in the retina, which are cells that control light sensitivity. Next, we have the CRX gene, which develops a cone rod homeobox protein that's found in the retina.
The next one is GUCY2D, a gene that controls protein in the retina that stimulates specialized pigments that sends a vision to the brain to be processed. Next is the IMPDH1 gene, which regulates cell growths in one's retina.
The next gene is the IQCB1, which encodes a necrosystem protein that interacts with the retinus pigmentosa GTPase regulator protein. In addition to that, the next gene is called the LCA5, which creates a protein that works with the splitting of a chromosome and keeps liquids moving on the surface of a cell.
The next gene is the LRAT gene, and it creates a protein that transports to the endoplasmic reticulum, which is a membrane that is in the eye that metabolizes vitamin A to use for eyesight. Next is the NMNAT1 gene. It encodes enzymes that help guide the nicotinamide adenine dinucleotide through biosynthesis.
Furthermore, the RD3 gene encodes a protein that is associated with the primolytic leukemia bodies located in the nucleus of cells. The next gene is the RDH12 gene, which encodes the protein NADPH, dependent retinol reductase, which is comprised of the enzyme that helps metabolize short-chain aldehydes.
Next is the RPE65 gene, and it produces a protein that builds a wall of cells around the retinal pigment epithelium. The cell wall brings nutrients to the retina and is essential for vision. In addition to that, the RPGRIP1 gene encodes a protein for photoreceptor cells that are essential for the health of cone and rod photoreceptor cells.
The next gene is called SPATA7, and it provides aid in cell maintenance and transports proteins. Finally, we have the TULP1 gene, and it encodes TULP proteins that are comprised of 200 amino acids.
In order to alter the genes of the person suffering from blindness, we'd go in and fix the mutations on the specific genes that are affected. CRISPR can be programmed to create healthy genes that can replace the LCA-causing mutated genes. CRISPR would move into the cells and replace all of the affected genes with the newly created genes. This can be performed on either a young child who has the disorder or an embryo. When performed on the young child, CRISPR can edit the genes responsible for LCA. The eyes are an immunoprivileged system, meaning that they will not inflame when new antigens are introduced to them. The mutated genes in the eye cells would be altered using CRISPR, allowing the child not to lose vision completely or possibly even bring it back after it has been lost. An embryo is a much easier system to alter. However, since it's only made up of one cell, as long as the mutated genes are corrected, the child will continue to develop and corrected genes will spread to the rest of their body cells.
Once the corrected genes are in the person's body, they will become part of their DNA. Because of this, their future offspring will be able to inherit the normal genes, putting an end to the spread of disorder in one's pedigree. Through sexual reproduction, the genes created by CRISPR would be passed on to any unborn offspring as they are part of the parent's DNA. From here, those offspring would then pass these genes, along with any new ones, onto their own offspring.
When CRISPR removes and replaces the genes that cause LCA, some people may be worried that it could cause many other problems. However, this is not the case. The genes that would be removed would be replaced with healthy, unaffected genes. This way, there would be no problems that could be caused by replacing the unhealthy genes, because they are still the same exact genes, just a version without the mutations.
While people might be concerned that editing genes might cause different issues, the result will outweigh the concerns. As a result, blindness will be cured and can be eradicated. While the procedure will be expensive because CRISPR will replace many genes if needed in the embryo or child, although this procedure may be costly at the moment, it will be well worth as LCA, along with any other disorder that can be fixed, would be stopped from spreading to future generations.
With the recent advancement in genetic alteration of CRISPR-Cas9, many diseases and disorders can now be put to an end. By replacing the mutated genes in one's DNA, they can be relieved of life-altering conditions that would otherwise have to endure. Using CRISPR, we believe that doctors can put an end to LCA, and hopefully eventually all forms of blindness. Thank you for watching our TED Talk.